Okay, well, it's Thursday again, so it's time to welcome you back to Science in the Age of COVID-19. Today, we are extremely excited to welcome Dr. Keith Jerome from the University of Washington and the Fred Hutchinson Cancer Center in Seattle. Keith is many things. He runs his own research lab. He's a virologist. The research focuses on persistent infections, such as, for instance, HIV, herpes simplex, hepatitis B, with a main focus of figuring out how these sneaky guys figured out how to evade immune surveillance and destruction. As a follow-up to that, if they can figure out how they're evading, then they can try to foil that. So they're investigating potential therapies based on gene editing, such as with CRISPR-Cas9, to harvest white blood cells from a patient with a chronic infection, fix them such that they're resistant, and then reintroduce them. And he's done a lot of other stuff on the side, such as looking at T-cell responses in various cancers and multiple sclerosis. He has other things as well. And what we will hear about today is his role as the head of virology at the University of Washington. Now, almost all the attendees probably know that Seattle was the place where honestly, the scientists on the ground, figured out what was going on, that COVID was here, that it had been here for a while, that it was spreading, and that we really had to get our act together. And so Keith will present us what he and his team have been doing since we figured out what was going on. So Keith, you are up.
All right, thank you very much. It's a pleasure to get to speak with you guys and tell you a little bit of the story about what's been happening here in Seattle over what's now coming up on almost on our 6-month anniversary of the virus being here in the US. And we've certainly been living with the virus globally now for a little over 6 months. So like any good card-carrying virologist, I have to start off with just a little bit about the virus itself. So SARS-CoV-2 is the virus that causes COVID-19. It is a member of a coronavirus family, and there are other ones, several of which we call endemic. That is, they circulate around year-round and cause things like the common cold. So these are viruses like 229E and NL63, OC43. And then there are other coronaviruses that get to be more serious. So you've probably heard about MERS, that's the Middle Eastern Respiratory Syndrome coronavirus that comes from camels and generally jumps directly from camels into people and causes a very severe illness with a lot of mortality. And then, of course, the original SARS from 2003, which genetically looks a lot like this one, but actually has quite a different presentation and was actually much, much easier for the world's public health efforts to actually control. All of these are positive sense single-stranded RNA viruses. Probably need to tell this group that simply means that the strand that's packaged in the virus can actually be translated into protein. It's a really big genome for a coronavirus or for an RNA virus in general. Think about RNA viruses. HIV has about 10,000 base pairs, hepatitis C about 10,000, hepatitis B only has about 3,000 base pairs. This is a big virus. And because of that, the virus actually doesn't have the luxury that a lot of RNA viruses do to have very low fidelity replication and create a bunch of quasi-species and mutation. And in fact, this virus uniquely has a proofreading enzyme and therefore has a fairly low rate of mutation. You'll see some of the work that we've done along with others to trace the outbreak by mutations. And actually the mutation rate in this virus is almost perfect for doing that, and that you don't have a huge quasi-species within a given individual. So you can actually track these as the virus spreads. There's 4 structural proteins. You'll hear me talk a little bit about spike. That's these little things that give the virus its name here on the surface. Those are highly involved in viral entry. And that's where neutralizing antibodies are actually going to bind and prevent the virus from infecting new cells. You'll also hear a little bit about the nucleocapsid protein that actually surrounds the viral genome. And we like that because it's highly immunogenic and a really nice way to diagnose the infection or past infection. And of course, this all was the cause of COVID-19, which got started in Wuhan now about 6 months ago. So we've been dealing with a lot of issues around diagnosis, and I spend professionally in normal times about a third of my effort doing viral diagnostics and improving them and improving their utilization, how they are used to manage patients and make things better. But this has been unique because we sort of suddenly out of nowhere had a new virus that we had essentially no diagnostics for, and then an unprecedented amount of demand. And so this is just a position piece that we put in that we published early in the outbreak, sort of illustrating this issue that you've got just a tremendous number of people at risk, most of whom actually are untested. There are people who are trying to get tests, but they're unable to actually get them because testing is too slow or it's inaccurate and all the problems. And actually only a subset of folks actually get an actionable result. And we heard a lot about these problems early on in the pandemic, and especially here in the US. We sort of got a break and thought we dealt with those, and now you're hearing those come up again, where places like Texas and so forth that are having new outbreaks actually can't get enough testing. This is now a week old, but the virus has really spread all throughout the United States, and you actually see more intensity here in the southern tier states now because of the activity of the last week. One thing that I always like to point out if I'm speaking to a hometown crew in the Pacific Northwest is the virus actually landed here first. And we're actually pretty small and underrepresented in infections, frankly. And to some degree, I think that has to do with the fairly effective public health effort that has occurred in a population that's been willing to take reasonable steps to control viral spread. And hopefully we played a little bit of a role in that as well by offering early diagnostics. So I'll start with, before I get into the diagnostics that we use routinely, I'd like to make the point that the virus, in my opinion, actually landed in a pretty good place because we were prepared as much as anybody in the United States for the virus to show up. And one thing that we've done for a long time is metagenomic sequencing. That is that if you have a patient with an unexplained illness and you do the regular virology test and you can't find them, you can't find bacterial, you can essentially just sequence a specimen from that patient and just look in an unbiased manner and compare that to databases of viral sequences and say, oh, well, it turns out it's virus X. And so we actually did that with some of the early cases. And the point of the slide, this was actually led by a student in our lab, Vikas Padu. And the point of this is simply to say that even before there was SARS coronavirus, if we simply took the results of our metagenomic sequencing and compared them to a pre-COVID database, we actually find something that spits out as a SARS-related coronavirus, not otherwise specified. And it turns out it would be the new one. And it's also highly related to coronaviruses that have been seen in bats and pangolins. So we'd instantly know we're dealing with something new. And if you do that, it's very, very sensitive. So here's all of our patients, and you see that here's 6, I think, of our early patients. Here's some negative controls, no reads on a coronavirus in any of these. And then you find other infections. Turns out, for example, this early patient also had coronavirus at the same time. The negative folks, why are they sick? Well, they're sick with coronavirus, typical common cold stuff. And then they have bacterial commensals that you might expect. So one great thing about this is that it really illustrates the necessity of having an unbiased approach should something actually start here in the United States rather than come from another place where we have a little bit of a head start. So we've done a lot of sequencing, and we've deposited close to 2,000 full viral genomes now. For a while, UW Virology was actually the largest depositor of genomes in the world. We've subsequently been passed, which is fine, but we put these into publicly available databases, and you can create relationships between viruses and start to see where they've come from. And so we've done a fair bit of work on that. because the virus really established itself here in the Pacific Northwest and the sort of Cascadia part of the continent early on, probably hit either here in Washington or just over the border in British Columbia sometime in early January. And what we saw is that essentially all of our early cases really clustered very tightly with an evidence of a second introduction event that actually didn't take off and seed our local area much at all. So this one sort of died out for the most part. But early on, all of our cases were actually this. And we worked with other folks around the country to look at that. And we worked with Charles Chu in California. And what they showed is that some of these cases from Washington or British Columbia, they actually got exported down to California. And so if you look, this is one of these early cruise ships that had a big outbreak, and you can see that some of these cruise ship isolates actually clustered together with the Washington isolates. Now, what was interesting and made us feel a little better that we weren't at complete fault about what happened in the country is that if you looked at the same time a little bit more broadly in California, what you saw is that California was having a number of introduction events really pretty quickly after we had seen the first cases. So although our cases did spread, they weren't really what seeded the entire country. And that's especially true if you looked over on the East Coast. We were doing, because we had such an early and robust sequencing capacity, we did a lot of, well, first of all, just diagnostic tests, but then also sequencing for folks in New Haven, and worked with this group in Yale, and basically showed that, yeah, the Washington strains actually did end up in New Haven and were a lot of the Connecticut ones. But really, what really happened on the East Coast was those were quickly overwhelmed by an enormous number of introductions, mostly from Europe. And so the New York outbreak, for example, was really multi-lineage, and the Washington strains were only a pretty small subset of those. So that's all nice and sort of the science of how things spread. But of course, for a lot of this, I've had to just take a lot of my basic science hat off for a while and put it away and just deal with diagnosis. And so we were really challenged as a nation with how to diagnose these things. And we were a little bit the victims of a bit of American exceptionalism, I'll say, because the World Health Organization adopted a test that had been designed by a group in Germany that actually performed quite well. So testing in this country actually got pretty quickly centered within the Centers for Disease Control. And that was actually kind of an unfortunate thing. In early January, when we heard about the cases occurring in Wuhan, we've done a few things right, but the biggest thing we did right in early January was we said, well, there's this odd respiratory illness in China. A lot of people seem to be getting sick. I think the Chinese have this under control. I don't think it's going to go anywhere. If it does, it's not going to come to the US and we'll stop it. But just in case, let's start building some capacity to do diagnostic testing. So we actually started a program to develop SARS-CoV-2 tests. So that was early January. And so really by the latter part of that month, we were ready to go with a couple of things. First of all, some primer and probe sets that we'd developed. And then we also saw the primer probe sets that the WHO had adopted. And so we evaluated those and felt that some of those actually performed better than what we had developed. So kind of made that our thing. And we're really ready to stand that up and stand fully prepared if virus showed up in the US. About that time, unfortunately, COVID-19 was declared an emergency. And I won't get into too much about the regulatory stuff about diagnostic virology because it would bore you to tears. But once something is declared an emergency, a laboratory like mine that is a very, very well-respected lab with a long history of developing diagnostic tests that are used for human diagnosis, for lots of other viruses. Once something is emergency, we're actually not allowed to do that anymore. And now everything we do has to be carefully scrutinized by FDA. So we spent a lot of time talking with FDA about what they would require in terms of documentation for us to offer our test clinically. They're not used to working with smaller academic labs go, we're huge, but compared to a multi-billion-dollar diagnostic manufacturer, we don't have staff and legions of lawyers to do all this stuff. So I got bogged down for a month just dealing with paperwork. And you might have seen some of the stories from our lab. We were actually forced to send a physical CD-ROM or thumb drive with our documentation rather than just email it to them, and they wouldn't look at it until those physically arrived. I mean, just crazy stuff like this. At the same time, of course, all the testing was being controlled at CDC, and then the test was flawed, and I'm sure you've heard that. So it actually didn't work, which meant that almost none of the public health laboratories around the country could do the testing, nor were academic labs like ours allowed to pitch in. I'll just point out, you guys are thought leaders in this. It's important for you to understand that we tend to think about public health labs as these behemoth labs, and they can do all this testing. Our situation is not that unusual. Our laboratory capacity is about 40 times what our state public health lab can do. And that's not unusual. State public health labs are really good at finding something new that's coming in, doing a broad variety of testing. But when thousands and tens of thousands of tests need to be done, they get overwhelmed. So it's very unfortunate that we were sort of shut out through what was really the critical period where we might have been able to control this more effectively. So finally, things got so out of hand that the FDA sort of relented, and basically about the time I think we nearly had all of their questions answered, they basically said, okay, academic labs can start to test as long as they've completed their validation and are basically themselves satisfied, and you have 2 weeks to get the final documents into us. And so we started. And so we started actually on March 2nd, and we were testing. At the time, we were the only place in Washington that could test. It one of the few places in the country actually offering tests. So it got pretty crazy up here. So what goes into all this? A lot of work goes into actually validating tests, and a lot of it's pretty, I don't know how to put this. It doesn't necessarily take the most creative amount of work sometimes to do this, but it's important and it's critical these tests work. So we did a lot of very careful, just multiple replicates once we finally had virus available to look at. This is the WHO primer probe set. Here's the CDC N1 and N2. Remember, they actually had a third one that didn't work at all, N3. It worked poorly. BGI is a Chinese company we looked at. We also looked at their own LDT and just seeing how they work. And there's actually differences in how sensitive they are. But generally, we determined that the N3 was terrible. The 1 and 2 were okay. The WHO were fine, as were these. So start to get some data out there that laboratories, remember, who at this point didn't have any idea what to do, could adopt these primers, that these were the ones they wanted to go in as they developed their own tests. Subsequently, of course, if you look at academic labs, hospital-based clinical labs, it's only a subset that really have the capability of making their own laboratory-developed tests and then validating them to a point that they can be used for patients. So what really gets the capacity of the country as a whole up is if companies come in and start manufacturing things. And so we had access and actually run a lot of these ourselves to have more throughput. So this Cobas is from Roche. I'm sure you guys have all heard of Roche. This Panther is from Hologic. Diasorin has a test. So we actually just compared then how they all work. And in fact, fortunately, these companies know what they're doing, and most of these tests work quite well. There are subtle differences in sensitivity, but most of the tests really work certainly well enough to be extremely useful in controlling and diagnosing the pandemic. The one exception you might have heard from this is there's this rapid test, this ID Now test that's really had some issues about sensitivity. Unfortunately, that's the one they use at the White House, of course, because it gives you answers quickly. It's been highly touted. There were some issues about what sample types go in there. Some really don't work well at all. With the optimal sample type, it's probably useful, but it still doesn't perform as well as a lot of these other tests. And then just a lot of work about what sort of samples can you use. So can you use sputum? Can you use plasma? Is there virus in CSF? What about stool? And which assays can actually each one of these be used on? How do the samples need to be handled before they get to the lab? Should we freeze them? Should we freeze them at -20? Should it be at -80? For how long? Is it better just to put them in the fridge at 4 degrees? For a lot of them, it is. It's actually better to not freeze thaw them, get a more sensitive assay on those tests. So these sorts of stuff. And then because laboratories started to become overwhelmed, in a lot of these laboratory developed tests, you would actually do 2 tests on each sample, the N1 test and the N2 test, for example, from CDC. So can you put those into the same reaction well and look at that? And all these were actually led by one of our students, Garrett Perchetti. So this kid has been hoping to get into medical school. He's actually going now, but a couple of the first author papers certainly didn't hurt his chances whatsoever. So he's done a tremendous amount of work. So I just want to call him out for that. And then I should mention, there's a nice test from Abbott. This is not the rapid test. This is their more high throughput laboratory test. This is actually, surprisingly enough, or maybe in contrast to what I just told you, actually probably the most sensitive test that's out there. Again, they all work really well, but for patients with really low viral loads, this would be the last one to be positive, even at low viral loads. One of the questions that came up early on, and you might remember this from articles in the lay press, a couple papers came out of China that said it looks like PCR testing actually has a lot of false negatives. That is, we're getting negative results, but it turns out that people are actually positive or we're missing it. One report said 30% of negative results are actually false, and the virus is actually there and the person's infected. And that was concerning to a lot of people, of course, because we're doing these tests and then telling people to go home, or we're telling them if they're in the hospital, they don't need to be in an isolation unit. It's actually very important. So we did a study, started a study, and then found out that one of our former trainees, who now leads the lab at Stanford, was doing almost the same thing. So we joined forces and actually published this together. And the idea was simply to look at patients who we tested, gave a negative result to, and then they went back into the community and then decided for whatever reason they needed to be tested again within a week. So they still have whatever their symptoms were. They still have reasons to be concerned. This self-selected population ended up looking at about 20,000 people over the 2 institutions, UW and Stanford. And the gist of it was that if you looked at people over the next week, only about 3% of these people who came back and felt like they needed another test, only about 3% of them were actually positive on the second test. So clearly we're not giving a ton of false negatives here, where the test is just flawed and then the second time it works. And of course, this is probably an upper bound because a lot of these people may have actually been infected, could have been infected during the interval between these. But so it gives you an upper bound that the tests are actually quite sensitive and very likely to pick up true positives. So we were really happy early on that we got testing going quite early in Seattle, and our state government was quite proactive in acting on the data we were providing them and so forth. So we had a lot of early social distancing put into place, early precautions. I don't really like the term shutting down, but gatherings were curtailed and many of the other things. And we actually were successful in flattening the curve. So here we started in March. These are test volumes, they vary. Weekends have always been lower. That's the gray bars. So how many are we doing in a day? But by any measure you look at, either Washington State outpatients, Seattle area outpatients, or emergency department patients, by all of those, our positivity rate, what proportion of tests we did were positive, actually peaked right around the end of the month. So gets up to about 12% for the outpatients, regardless of where you look. Maybe a little bit higher for the emergency department patients, people who are truly, really sick. And then it really fell down quite a bit after that. And we actually worked with our stats folks and projected this out and sort of felt like by the middle of May, if trends continued, we'd be down in the 1% to 2% range. That actually got taken out of the paper at the behest of reviewers, but turned out to be right. And by the middle of May, we were running 1.5, 1.8% positivity rate. So pretty much a success story, and maybe not so much different in pattern, although much different in magnitude from things that have happened, for example, in New York, where the number of cases and the positivity rates have dropped tremendously. We've done a lot of work to support our clinicians in doing testing. So I won't go into this too much, but this is a paper of our early results, just caring for patients. And frankly, people didn't do very well. So the early patients, this is the first series of 24, and half actually ended up dying. I think this was pretty common experience early on. People didn't get into the hospital quickly, and our clinicians were learning how to care for the patients. Things have gotten better. This is more recent experience, and now the mortality rate's substantially lower, and many more people are getting discharged than are dying in the hospital. But this is still a major problem, and particularly under situations where the healthcare system is stressed and there just aren't enough resources to actually take optimal care of all the patients. And I fear we may be getting back to that situation in some parts of the country. We've been screening all the patients now in UW Medicine. So this is started in our obstetrics unit, and we tested everybody who was coming in for a procedure or to deliver birth, and tested quite a large number of people just to find 9 folks, and they were truly positive. For most of them, we detected a second time before they went negative. A few seemed to have a low viral load and went negative pretty quickly. And we've subsequently expanded that to all University of Washington hospital system patients. So everybody comes in for any admission or for anything that'll generate an aerosol, they all get tested. We've done a lot more than this now, but this is what was in the paper. And in any of these screenings, the yield is about 0.5%. So at least during this time, asymptomatic or minimally symptomatic patients who were coming up for other reasons, about 1 in 500 were actually positive. So one way to look is this awful lot of testing for not much benefit, but actually you need to think about the catastrophe it would be if these people got admitted to the hospital with undetected COVID and actually then were able to spread it to other vulnerable populations in the hospital. So this is well worth it, and we feel we've probably prevented 20 to 30 hospital-associated cases by doing this. We've evaluated the prevalence of infection among our employees. We opened a drive-in employee clinic very early on. So if you were an employee of our hospital system and had reason to think you might have COVID, either an exposure or symptoms, come on in. And overall, the sort of overall prevalence has been about 5% over that time. So this is, again, kind of symptomatic, self-selected people, but it's very important to then get these caregivers the care they themselves need, and also to keep them away from our patients. One thing I was really, really proud of, and Josh Lieberman, who's actually sort of on loan from our bacteriology part of our department, came over and helped us and led this effort, which was basically to ensure a very rapid turnaround workflow for prospective organ donors. And the issue here was there's concern that one could spread the infection via organ transplantation, tissue transplantation, to an immunosuppressed population who presumably wouldn't do well with that. And just the graph here just essentially shows what proportion of people who are waiting for an organ actually get put on an inactivation list. So essentially, or is the wait list itself inactivated? And if you looked early on in the COVID epidemic, in various parts of the region, essentially these transplant services shut down. That 80% of them just said, we can't do transplants now. And this actually, these shutdowns actually persisted, but I was very proud of the fact that here in the Pacific Northwest, because of this rapid testing protocol, within a week, we were able to essentially open those waiting lists right back up again so that people could get the organs that they needed. So a real win. In this gets a little bit into the delivery of clinical testing aspect of things, but it's not only important to be able to do a lot of tests, and we do a lot of tests, but it's also important to be able to do tests rapidly. You've heard these stories about people waiting a week or 2 weeks for their tests. That doesn't work if you're waiting for an organ transplant or really anything else. And so we've really prided ourselves on being really rapid about getting results. We have done studies in group living facilities with colleagues here at the university, and this is a really nice early illustration that by going into group living facilities, nursing homes, assisted living facilities, correctional institutions, and intensively testing, you find cases and you can prevent outbreaks. And so this was a local nursing home that we went into very early on, and essentially tested everybody. And we found people who had really no or minimal symptoms, either residents or staff members, and we're able to test them, find out they were positive, and then get them separated from the rest of the population. And this prevented any outbreak in this facility, actually. So the facility had no more cases after this. And this has actually really changed government policy here in the state. In fact, we just completed, and I might have this on the next slide. Yeah, it's just a little out of date, but essentially, the state then required all these health facilities to begin routine testing. So these are the number of tests we do per day. And because we're the biggest provider in the state, a huge proportion of them came to us. And you can actually see when this jumped up is when this went into effect. And a lot of these are simply keeping people in these areas safe. So this is our Twitter feed, by the way. If you're interested in what's going on in the Northwest, let's go to hashtag UW Virology or at UW Virology, and you can see where we are. We're typically doing about 5,000 tests a day now. Our positivity rate has crept up. For a while, we were down around, as I mentioned, this 1.5% to 2%, sort of in this range in mid-May. We've started to bump up in this 3. We've had a 4% day. That has us concerned about what may be coming here in Western Washington. And if you look in places like Florida and Texas, they're having positivity rates in certain areas of 15, 20 or more %. So that's a bad sign when those rates go up. I want to change gears a little bit and talk about serologic assays. So everything I've been talking to you so far is looking for the virus itself. Is a person infected right now? Antibody testing is different, right? Our bodies make antibodies in response to infections, and that's how we fight them off. And I'll show you a little bit of data that clearly shows that. But essentially, testing for antibody then gives you this sort of historic record of has someone been infected in the past. This can be used for population-based studies of seroprevalence. How many people in your area, in your city, in your state have been infected with COVID? This in turn can inform public policy decisions and recommendations. Occasionally can be used in primary diagnosis, not very useful. I get asked a lot, if I have these antibodies, does this mean I'm safe from the infection? The jury's still out on that to some degree. I'm pretty confident that they're going to give you some degree of protection. It may not be complete. In fact, it's probably not complete. And I'll bet you that people with these antibodies can still get infected. The most likely outcome, though, is that they don't get nearly as sick. They don't shed nearly as much virus, much less likely to end up in the hospital with severe disease. But from what we know about previous coronaviruses, protection is not absolute, and unfortunately, nor is it lifelong. It actually seems to wane within a year or two. And I'll just mention in passing while we're here, we're starting to see signs that these antibody levels actually do decrease in people a bit more rapidly than we might have hoped. So when we were looking for a serologic assay, we wanted a couple of things. Early on, you might remember sort of FDA was so careful, overly rigorous about regulating the molecular testing for the virus itself and caught a lot of flack and sort of, I think, overcorrected and basically said there was no regulation on serologic tests. So very early on, the vacuum of testing was filled with really terrible tests that had very, very poor sensitivity and very poor specificity. And so there was some bad press around, you just can't believe these antibody tests. So we wanted something that was good at detecting the virus, and especially important, it was very, very specific. The last thing, so specificity in simpler terms just means if I give you a positive result, how confident am I that that's true? It's not a false positive. And that's important because people are going to make decisions based on this, right? Potentially, particularly if we find out that you're protected, we don't want to give you a false sense of security. So specificity was critical. Of course, we wanted to correlate with meaningful immunity. And then we wanted to be able to do a lot of these, right? So ideally, you'd go on something we had. So we ended up choosing an assay from Abbott to evaluate. So we had access to this early before it was actually released. We've worked with Abbott for on various sorts of projects over years. We're often a laboratory that manufacturers will go to for sort of an independent validation that something they think works actually does in somebody else's hands. So we got access to this. It's a chemiluminescent microparticle immunoassay. Qualitatively detects the virus. I'll show you there is some quantitative information in it. It detects antibodies to the nucleocapsid. So the nucleocapsid, if you remember that picture I showed right at the beginning, is on the inside of the virus. So these antibodies are not likely to be the kind that can bind to the virus and keep it from infecting new cells. So they're not going to be neutralizing. But I'll show you they actually correlate with the meaningful immune response. So because it's so immunogenic, it tends to be a very sensitive assay. So it's very, very good for these purposes. It occurs on it's performed on a common analyzer that we have several of now in lab and that actually are common around the country. So it is something that can be provided broadly if demand is there. So we evaluated this independently again, just how does it perform? And so the first thing we did was just to take patients who were admitted into the University of Washington system, so hospitalized, they're sick with COVID, and ask what do their antibodies look like at times after they've had their onset of symptoms. The assay actually returns you something called an index value. It's a rough proxy for how much antibodies there. And essentially, if this value is above 1.4, according to the manufacturer, it's positive. So anything above the red line is positive, anything below is negative. So you can see this is all going up, either at time post-symptom onset, or you can do it days after your first PCR positive. Positive test will always trail symptoms generally in these folks. This is why they got the test, because they had symptoms. And then you can plot that, and you can see that we achieved 100% sensitivity. Everybody who had COVID had antibodies by 17 days after their symptoms started. Okay, so very, very sensitive test. So that's nice performance. And then maybe more importantly, what's the specificity? So we do a lot of testing for other viruses. We took a sample of, or a series of over 1,000 samples who had been sent to us for something called the herpes simplex virus Western blot. This is the gold standard assay really for the world for HSV serology. So difficult cases are sent to us for this, but these were cases that all came in before there was COVID, so we knew that they should be negative. And so out of 1,020, we found one that was a false positive with an index value of 1.41, so 1/100th of a point above that. So a very, very specific test, 99.9% specific. Abbott had generated a number of 99.6 in their hands, so we were a little bit better than them, and that's probably just statistical noise. And in fact, then we could set our own cutoff based on everything we'd seen of actually between 1.42 and 1.49. And if you use a cutoff anywhere in there, you actually get a perfect test. In laboratory medicine, we do these things called receiver operating characteristic curves, where you basically plot the specificity against the sensitivity at any given cutoff. And usually there's a trade-off of these, and you get some sort of curve. And the more it looks like a triangle, the better, but it never does in my 30 years of doing this until this came through, where we actually had a perfect test that was 100% sensitive and specific using our own cutoffs. So anyway, really, really great test and very useful. One kind of cool thing is that we evaluated, we always want to evaluate in a test, how reproducible is it? If I give you a result, or especially if I give you a number, how close is that number to the number I'd give you a second time? And so if you plot just the index value, so we took patients who had more than one test done in a single day. So I figured those ought to be the same, right? So we have these different samples, and then we plot them. So not surprisingly, we see this a lot. If assays are sort of higher in their measurement range, they tend to be more precise. So the coefficient of variation is pretty tight. And then as we get lower, there's more variation. And then we noticed that there were these right around the cutoff that were kind of unique. They had a lot of coefficient variation. And we looked at those a little closer. And in fact, what you see is that this poor reproducibility is actually biologic. It's not something about the assay. And it is these patients who had multiple samples taken over the same day. These were actually taken on the day they're seroconverting. So if you plot the day of symptom onset, and to the hour here, you can actually watch patients seroconvert during their hospitalization. So here's a patient and they're negative, and then suddenly here on day one, boom, they go positive. Here's one who was seronegative for the first 2 weeks they were in the hospital. And when they finally seroconvert, look at that. You can see that all happened within a day. So really, really interesting test and useful also in terms of studying these early events as the body actually develops an immune response to the virus. We've had a lot of concern around correlation of this assay with the protective immunity, which is expected to be antibodies toward the spike. There's actually a receptor binding domain within the spike that you can even look more carefully. But you can compare the Abbott assay, the nucleoprotein assay, from a different manufacturer an assay from Euroimmune that targets the spike. You can see they actually correlate quite nicely. This is sort of correlation that we're usually pretty happy with in laboratory testing. Everything that's positive by one is positive by the other. They might disagree a little bit on the quantitative amounts, but again, neither one of these tests is marketed as a quantitative test. It's actually quite good. So we think that if you see something positive here, if we scientifically find that being seropositive does have a protective effect, we'll be able to extrapolate from the Abbott assay. Here's a study that we did together with folks in Idaho. This has been a little while ago now, so pretty early. Idaho had an interesting outbreak, very much centered on an area around Sun Valley, which is a big ski resort and place where a lot of CEOs and wealthy folks have places, a beautiful area in the mountains. So turned out there's a lot of air travel into there. I think that's where Tom Hanks went when after he was diagnosed and got home from Australia. I'm guessing he was there in Sun Valley in his house. And so we looked at the closest major city, which was Boise, and just asked people, hey, do you want to be in this study? They signed up online and came in. So kind of a self-selected population of folks who probably had some reason to want this test, thought they'd been exposed, maybe they had some symptoms at some point, and actually didn't find too many positives. So about 1.8% of close to 5,000 patients, and pretty broadly distributed in age. Maybe a little peak here in the older populations, but hard to say. And as you break this down, the stats start to fall apart. We've tested a lot of patients now. This is a little out of date, probably done about 40,000. And of everybody who sends in their test to us for whatever reason they wanted, about 5% have been positive. We've done other sorts of seroprevalence testing. So here's an updated number on our clinical testing, still a bit out of date. We've tested patients in UW medicine, so just our internal patients, they're a little bit higher. I spent half my time at the Fred Hutch. That's where my basic science work is. And we did a return to work study. The Hutch was essentially shut down for no research for about 6 weeks. And as we started to bring people back, we randomly selected people to participate in the study where they got viral swabs and serologies. So our population is a highly educated, relatively younger population as a whole in the Seattle area. And of these folks who had been self-sequestering quite well, the physical distancing, they'd be expected to be pretty compliant to all that. About 1.2% were positive. We're now doing a big study on UW Medicine employees, looking at all the employees in UW Medicine. We just got some results in from our frontline workers, so the ones in the units taking care of COVID patients and so forth. And the positivity rates there don't look any higher than they do in our population here as a whole, which is encouraging. It means that we're able to keep our healthcare workers safe, the personal protective gear is appropriate, and is actually working. So we're happy about that. But of course, all of these are sort of self-selected in some way. So we just have been approved for a really nice statewide seroprevalence study. This is funded by the Paul G. Allen Family Foundation, and it's in partnership with the state and then the university. And we'll be looking at 7,000 participants, and they'll all get an initial PCR test and then serologic testing, follow up at actually 4 and 8 months. And so what we'll be looking at is the geographic distribution of infections around our state, the distribution within potentially marginalized populations, so minority and ethnic populations, people in certain high-risk employment groups and so forth, to get a really broad sense about what's going on right now in Washington, what's going on in the fall as we move into the higher risk months where influenza is likely to come back and other respiratory infections, and then in the spring to see where we stand. So we're really excited about getting that started. here in the next week or two. So just as we kind of come to the end, I want to show you one bit of data that I've been showing to groups just to make a point is per the US FDA, for none of the testing that any lab like mine can do, all we're allowed to report is for a virologic test, is the virus there, a yes, no answer. So it's there, yes, it's not, no. Same thing for antibodies. But in fact, if you look at things and again, sort of look at viral load and it compares well with antibodies over time, but you can actually watch this sort of inverse relationship of development of antibodies with viral load. So the proxy for viral load is the threshold cycle for our PCR assay. So it's sort of inverse. This means low amounts of virus. If it's up here around these long threshold cycles, you have to do a lot of rounds of PCR to see the virus. That's basically what that means. 40 cycles of PCR to amplify the virus, enough to see it. And so as antibodies come up, generally people's viral loads are falling. But what's really, really fascinating is invariably, nearly invariably, the highest viral load we ever see in a patient is the first test we do on them. That is, and this is why this virus transmits so effectively, is the big viral loads are before people are sick, or at least sick enough to come in to the healthcare system. And once they're at that point, they're actually already getting better in terms of the virus. Now the immune response comes up and causes the lung damage and a lot of other things we're seeing. So that's part of the pathogenesis. But that early viral load is the highest one. So if you look at how much viral load people have at the time of their first interaction with the healthcare system, so people with these low CTs have very, very high viral load. So here's the CT on the x-axis. You can look at length of stay, which has some information to it, but let's just look at outcome. Did they die in the hospital? That's the x-axis. And you can see that there's about a fourfold increased odds ratio of actually dying if you have a high viral load, here defined as a CT less than 22. There's actually also a power of prognosis if you're seropositive at the time you show up at the hospital. If you already have the antibodies, so you're over here, you're actually likely to do well. If you're that sick but you already have antibodies, you're going to do better. If you're already that sick and your body hasn't made the antibodies yet, you're much more likely to go on to severe disease or be at risk of dying. So I think this is just a way of saying laboratories actually have the capability of giving our clinicians more information that I think would actually be useful in managing and stratifying patients. And it's unfortunate that we're still prohibited from actually sharing that with the people who could use it. So what's going to happen now? And gosh, I think most of these slides, I'll confess, are about a week old. And so the future, much of the future is now. I said the demand for testing is likely to increase as economic activity resumes, and if we see more infections, and I think it's very clear now that we're seeing that, certainly in parts of the country, and we're very concerned here as well, and I think frankly we should be again in New York and other parts where the virus can certainly come back. I think we're going to get data that having these antibodies gives you at least some protection, and I continue to believe that's going to increase the demand for serology as people just demand to know, and we're prepared for that. The therapeutics, the pipeline here is uncertain. We've seen some nice work, some nice data now with steroid therapy to sort of tamp down this immunopathogenic response that we see late in the infection. We also know that remdesivir is good early on for reducing viral load and likely getting patients out of the hospital sooner, probably saving lives, although the study wasn't powered to be able to show that. So I think that some combination of early control of virus and then control, if needed, of an overexuberant immune response is likely to be useful. But I'm not sure there's going to be any magic bullet coming through that pipeline in the near future. One bit of good news is that the vaccine effort is actually quite well organized and is moving ahead quite well. There's a number of candidate vaccines. The first one from Moderna will be starting its phase 3 clinical trial, so a big efficacy trial with 30,000 participants, scheduled to start July 9th, still seems to be on course for that. We'll be doing a lot of the virology for that study. Not too far after that is a vaccine from AstraZeneca, probably 3 or 4 weeks behind, and then several other vaccine candidates will be ready by late summer. So as Fauci's gotten on the TV and said, there's a solid chance we'll have a vaccine approved and starting to become available by the early part of 2021, and just maybe we might even get it late this year, which is really a remarkable feat if we can pull that off, and will be the one time I think we'll have something to kind of be proud of is what we've done, because the country's not really handled this pandemic particularly well. So I think I'm probably coming close to my time, so I won't say all of these things again. Couldn't even acknowledge all the people. I tried to put up all the names as we went. One thing you've really seen from this is it's unbelievable, the new collaborations and the degree to which people are working together on this. There's so many new co-authors on papers that I never had before, and so much great collaborative work. But it's been a real tremendous effort, and we're very excited to continue to work on this and hopefully turn the tables on the virus with the vaccine. So with that, I'll stop. Thanks for your attention, and if there are questions, I'm happy to take them.
All right, well, Keith, that was spectacular, and we do have a lot of questions. Today we are rolling out a new way of people being able to ask their own questions. Wyatt, you are first up to ask your question.
Great, thank you. Thank you very much for the talk. It was very informative. My question is about the SCAN, or Seattle Coronavirus Assessment Network study. I'm curious whether there are any plans you're aware of to expand that study, perhaps even outside of Seattle. That's the first part. And the second part of the question is, what can we learn based on this study about true prevalence in the community? Is there anything we can infer about actual numbers, or do you think that the, say, the 2 to 5% is what we should be using for these models?
Yeah, those are great questions. So the scans struggled a little bit, unfortunately, with some of the issues around some of the regulatory issues, quite frankly, and those were in the newspaper and so forth, and FDA. One of the issues is that as a research study, some of the requirements weren't in place to be able to report things back to individual patients. So a lot of those have been worked through. We know the investigators very carefully. So we've gotten through that. In terms of scaling that up, I think, quite frankly, it's pretty overwhelming what we're trying to do here. I think that that's a great model. I know those investigators are interested in working with others to sort of replicate that, but I'm not sure that it takes a lot of effort to do that and a lot of kind of feet on the ground. I think those numbers are probably about right. I think that they're low, and I think that 2% is probably, it's a moving target, right? The beautiful thing about how they're doing this is that it's self-sampling, which means you can actually get people to do it themselves. Unfortunately, FDA doesn't believe in that people can do this effectively and reproducibly, so you can't actually report those back to patients. A patient can take the sample themselves, but it has to be witnessed by a healthcare provider. So it makes it, doing a study like that broadly, much more resource intensive. So in our statewide study, there is that sort of look, but we'll actually be picking people randomly based on census tracts and actually going out there with field teams and actually getting those and watching the samples be taken. So I think their numbers are pretty good, and I think that's, but remember, what we see today can be completely different. The doubling time of this virus, the potential doubling time of this virus is unbelievably rapid.
Okay, next up we have a question from Darcy. And I forgot to mention, so attendees, if you have a question, please click the raise hand button at the bottom, and it makes it possible for me to activate you. So next up is Darcy.
Great talk. So I think you've answered part of this. Very early on in the virus, a group of people at Columbia decided that really who were dying to get back to work, basically, decided that really the only effective thing that we could do that would make everybody feel safe was to develop a home test for the virus. So this group had, together with a guy called Chris Mason, who's at Cornell, and a bunch of international collaborators, have been working on developing a lamp-based saliva test, which, as you can imagine, is very ditzy. Saliva is kind of difficult, and it's a colorimetric test, and reproducibility and sensitivity and so forth are issues. So it's right now a research test. And what I'm wondering is, are the barriers to developing a home test sufficiently high that it would be impossible to roll something out like this, given the FDA aversion to people doing this at home? This is, you know, one of the goals of this is to make it idiot-proof, right? I mean, you don't have to know what a microliter is. And there have been some good engineering aspects to this. But it sounds to me, from what you say about the FDA, that getting this rolled out on a population level, even if it were working very well, would have huge numbers of hurdles.
Yeah, well, I don't want to be too hard on FDA. I mean, they provide a critical service in terms of making sure that tests do work well. And it's a challenge when you have this just emerging, exploding pandemic like this to react quickly enough and change things. So there are barriers to these home tests, and we've been involved in some work. I didn't talk about any of those. I mean, some of the Cas12b based detection systems and LAMP and some other things. Generally, what we're seeing is that the sensitivity is a challenge to get that to where the more mainstream tests are. And then you use the term idiot proof, but to make these so that they can be used successfully by people who aren't trained in diagnostic medicine. So something akin to a pregnancy test or an at-home HIV serology test. I think that's the goal. I think if we can achieve that and show that they're reproducible, that the average person can do them successfully, I don't think there's any reason to think that they won't get through the regulatory process, even if they're not quite as sensitive as the gold standard laboratory-based tests. They could still be really useful, right? I personally would rather have everybody in the country doing a not quite as good test every day and hopefully sort of explain the limitations of that. But I think we'd catch a lot more people than we'd miss. And that's the sort of case that we'll have to make as scientists in this field to the regulators, that the overall health of the country is benefited. Maybe I'm foolishly optimistic, but I think if the tests perform, we'll succeed at that.
Okay, next up we're going to have Adam ask a question, and may I ask Kim and Brenda, please raise your hands. Okay, Adam, you're up.
Hi, Keith. Great talk and great work over the last few months. I have two questions, actually. One question, one important matter is to try to figure out who is still producing virus that could be infectious. It's important for releasing people from the hospital, going back to work. There's been some early reports that a combination of RT-PCR and serology studies can be used to basically assess whether a person's still producing infectious virus and then therefore not requiring neutralizing tests. And I was wondering what your opinions about that were.
Yeah, so we know from a lot of tests that from a lot of viruses that generally infectivity is inversely correlated with viral load. So if you have not much virus, you're less infectious, and that holds for almost every virus we've ever looked at. And we've had this problem, right, that some people want to get out of the hospital and they keep shedding the little bit of virus that we keep seeing by the PCR. And so maybe we're keeping them in there unnecessarily. So some groups have done culture, for example, on those and said, well, look, you're not finding culture. The culture is negative, and I've heard people interpret that to say, well, that means that what you're detecting is dead virus. You have to understand that the sensitivity of culture is much, much less than PCR. So it just means there's less there. You can't say it means it's dead. One thing we've been doing is specific PCRs for subgenomic RNA species. These are odd little byproducts of the weird way that the coronavirus is actually translate their genome, that they actually make these odd, they look like spliced RNA. It's actually not. It's an odd stuttering phenomenon that makes them, but those only occur in the process of active replication of virus, and they're not actually packaged into the virion. So you can test for that. It seems to be kind of somewhere between, in sensitivity, somewhere between the regular PCR and the culture testing. But I think what we really need is just a clinical experience to say, yeah, people with these low viral loads don't transmit to other people. And that takes time, and you just can't generate that sort of data in 5 months. That's why it's still not clear what to do in these cases.
And one other question, for these sort of long hauler people who have really symptoms and PCR positive for long periods of time, have you sort of mapped through their serology over those periods of time?
Some of those people, yeah, I'm not sure we have a complete handle on it. Some of them don't seem to, are not seroconverting strongly, so that could be part of their issue. And so this kind of goes on and on and on. You might have seen a couple of those tracings that I showed quickly. Some people really have a delayed antibody response, and so for whatever reason, their immune system is not very robust. So that can be part of it over time. And so, like you suggest, maybe some combination of virus below a certain level and then antibodies above a certain level might, with a little more experience, give us the confidence to get these folks out of the hospital.
All right, next up we have Charles with a question. Okay, Charles, you are ready to go. Charles? Okay, Charles, okay, we're gonna let you figure out your audio issues. Kim, you are up.
Hi, thanks for the great talk. And you touched on this a little bit as people are heading back to work and society at large is trying to get back to work. Testing is clearly the most sensitive way of detecting infected individuals, which will allow us to isolate and do contact tracing. However, there is still the problem with test availability, the cost of those tests, and problems with frequent sampling. In your opinion, how would you weigh frequency of testing? Like, what's the minimal amount of testing that you would think would be useful, and what would be optimal?
So I like the idea of doing enough tests so that your positivity rate is below 1%. And we almost got there in Western Washington, and then I think things have started to turn. But what that just sort of implies is you're doing so many tests that are negative that your threshold for testing is so low, you start to get more likely to catch cases early on. Clearly, once you're up around 8 or 10 or 12%, you're really not testing until people are sick and the horse is out of the barn. So that's sort of the metric that we were shooting for in building capacity, working with the city to open more drive-through testing sites, for example, and that was part of the bump we've seen recently as those have opened up. That's the sort of thing that needs to happen. But now that you've seen, we've sort of, I think, again, missed the chance to actually keep the virus down under control. What it would take to get back to that situation now is going to be heroic, and I'm not sure that people are actually going to tolerate those sorts of restrictions again. So I'm not completely clear where we're going in the next couple of weeks here.
So do you feel like if employers could test on a daily basis, obviously that would be useful. And I think, as you mentioned, you wouldn't have to use a test that is 99% sensitive, right? You could testing somebody with something that's 50%, but every day is better than.
Yeah, but it's surprising how easily something like that can break down. So I'll give an example, and this is all public stuff. So there's, I'll just tell you what you can read in the newspapers. So we were doing testing for some of the Alaskan fishing fleets. So these people go out, you've seen movies about, they all get on a ship and they work 20 hours a day for 3 or 4 months, and they make all their money for the year in that time. And so they were very, very concerned because, of course, it's very closed environments. There's not a ton of medical staff. If they have to put into port, it's a little town on the coast of Alaska that doesn't want them, first of all, and doesn't have the hospitals to help them. So we did a bunch of screening for those. They were counseled by physicians in our group and others to do a test, isolate themselves for 2 weeks, do another test, and then if everything was negative, then at the end of all that, you'd have 2 viral tests plus an antibody test in there just to see what was going on. If everything was negative, you go straight on the ship and go. And a couple of the ships still had enormous outbreaks, and because they didn't. One company didn't want to pay to do the second test a week later. Some people weren't staying in their quarantine hotel, which was provided at no cost. It's human nature got in the way. So none of these things are going to be perfect. And even testing every day, you don't become positive. Unfortunately, you don't become positive instantly after exposure. And ideally, you can catch somebody at least before they're highly infectious if you did that. So maybe there'd be some success, but I worry that still people fall through the cracks. And frankly, we've talked with employers. There are employers here in Washington State and Seattle area who are quite wealthy employers, and there's not a lot of enthusiasm for testing every employee every day because that adds up to a lot of money pretty quickly.
I'm gonna maybe re-ask Kim's question in a less politically correct fashion. If an employer, such as a biomedical institute, was capable of putting together a good testing program, do we have a moral imperative to at least try? It's not going to be perfect. We're going to miss some things. We're going to screw some things up, but, should we try?
So I have not counseled the Fred Hutch to do that. I mean, they talk to me a lot, although it's my university hat for the diagnostic stuff, but they've asked me a lot, what should we do? And early on, we considered testing everybody once a week. And when you look at the budgets of that, I'm not sure it's actually necessary. Quite frankly, at least here right now, everybody who comes on campus wears a mask. Everybody does a symptom check every day. There are no meetings over 5 people. We have not had any workplace associated illnesses. I think with a highly motivated and intelligent workforce, you can actually keep things safe and do your job. And I think probably just destroying it's a lot of money to do a lot of tests. I mean, each one of these it's going to be a lot. And I think it's jaw dropping to your CFO once you start actually talking about this.
Yeah. I would just say it's probably more expensive to have employees die, but at that point, I will let that go, and we will move on to Charles, who has figured out his audio issues. So, Brenda, I clicked on you instead. So Brenda, can you go now? Okay, I'm going to click on Charles again. Okay.
Hello, can you hear me?
Yes.
Okay. Thanks for the talk. My question was kind of references back to the early part of your research earlier this year when you were sequencing patients to try to trace the lineage of the different infections. So, on the West Coast and on the East Coast, you were able to determine how much was community transmission versus, imported COVID-19. And I'm curious from the perspective of, let's say now looking at the sort of global perspective, when we see this sort of tiered viral response where, it started in Wuhan and then it moved to Europe and North America. And now it seems like places like Latin America, Brazil, North Africa and Southeast Asia, especially India, seem to be just about to be hitting the exponential part of their curve. Do we have any sequencing data from those regions, from those patients, to try to analyze essentially the same thing you did within the United States to try to determine where the infections are coming from and what kind of community transmission there is? Because it seems like there's still a lot of things we don't know about the epidemiology of this virus and this disease to explain some of the global patterns that we see.
Yeah, so So we have some information. It's very, very difficult to get primary samples out of most of the countries that you've named. There's just a sense that sending them to the US or other places is giving something up. So sequencing has trailed pretty severely, particularly in India, for example, and Brazil to some degree, of what you would expect based on the number of infections. But I think what we're really seeing now is that there's so much virus in the world that essentially when you see at a national scale, essentially what you start to see is just multiple introductions. And we had this situation early on in the US where there was that initial first introduction, right? And there were just worldwide wasn't that much. And it was probably, really looks like it was from Wuhan. But then you saw other introduction events in other parts of the country. And so things becomes this sort of mutt of viruses. I think the next area where the viral sequence is going to come to the fore, the next batch of important papers you'll see will actually be tracing very, very local outbreaks. That is, if there are high profile events with lots of participants, for example, where there's concern about holding something and should this be held, and you see infections, actually you're going to be able to say, yeah, those are all related. But in terms of country levels, there's still so much movement between countries, even though we think it's cut down, and there's so much virus out there that what you're seeing is multiple introductions into these countries. Hey guys, I'm really sorry. I'm going to need to cut this off. My lab is starting to text me all the time. We're having a lab meeting, and I'm late. So apologies, but it's been a pleasure speaking with all you guys. So thank you so much for the invitation.
Well, thanks so much, Keith. Okay, go save the Pacific Northwest.
Oh, this is gene therapy, actually. I'm going to get a break from COVID for a little while.
Okay, fantastic. And we really appreciate your time.
All right, take care, everyone. Goodbye.
Okay, bye.